Good morning, everyone. Today we are presenting our research poster on formulation of diabetic-friendly jelly bears using guava for blood sugar level control. We will briefly discuss our introduction, methodology, results and discussions, and our conclusions.
To start off, we have the introduction. Here we can see the background of the study and the purpose statement. So first is the background of the study, where it states that diabetes is a long-term condition caused by high blood sugar levels. It affects over 422 million people worldwide. Guava, a tropical fruit with health benefits, may help manage blood sugar levels due to its high fiber content and low glycemic index. This research aims to create diabetic-friendly jelly bears as a natural alternative snack, potentially improving blood sugar control and increasing consumer satisfaction with healthier food choices. So this section explains the problem or what diabetes is, and we present guava as a natural solution. It provides the context for why the research is important. And then we have the purpose statement. So this study aims to develop diabetic-friendly jelly bears using guava fruit, evaluating glucose release through in vitro digestion and God pod methods to provide a healthier snack option for diabetes patients. This clearly outlines what the study aims to achieve. It creates and evaluates the guava jelly bears as a healthier snack option, and this guides the readers to understand the research objective or our research objective.
For our research questions, we have: what is the average glucose level concentration of the different guava jelly bears? Second, what is the rate of the glucose release between different formulations of the guava jelly bears in terms of oral phase, gastric phase, and intestinal phase? Lastly, what is the pH range of the different formulations of jelly bears?
We have also included our conceptual framework to this poster to visually represent the study's process and key elements. So it serves as a structured guide that explains how the different variables interact within our research.
To determine whether the consumption of jelly products developed reduces blood sugar levels, the researchers used a quantitative method using an experimental research design that focuses on the formulation and production of sugar-free vegan jelly bears. The researchers analyzed the digestion and glucose release using the in vitro digestion method and measured blood sugar impact with the glucose oxidase peroxidase or GOD-POD method. These methods were carefully selected to provide precise insights into the formulation's effectiveness in blood sugar control. The researchers chose this method because it allows the researchers to collect and validate the data needed from the formulas.
A descriptive analysis was conducted to compare glucose release across three jelly bears formulations, A, B, and C, during in vitro digestion. The study used the glucose oxidase peroxidase known as God-POD method to measure glucose release across oral, gastric, and intestinal phases. Enzyme breakdown and pH levels were monitored to assess digestion conditions. Glucose was extracted and quantified from 10 jelly bears per formulation, while pH levels were measured from 1 jelly bear per formulation at each phase. Additionally, the average glucose concentration was determined by extracting a small sample from 1 jelly bear per formulation and analyzing it using the God-POD method. The findings provided insights into glucose release patterns, supporting the potential of these jelly bears in diabetic glucose management.
The research instruments or material section we have included in this poster is to highlight the tools used in measuring key variables that ensures the accuracy and reliability of our study's findings. So each instrument plays a crucial role in data collection. So first we have the colorimeter. It is used to measure glucose concentration in the jelly bears by analyzing the color intensity changes. So it ensures precise quantification of sugar levels. Then we have the magnetic stirrer. So this is an alternative for an incubator. So this helps maintain the consistency in the sample solutions by ensuring uniform mixing during glucose testing, preventing inaccurate readings. Next, we have the pH meter. This determines the pH levels of the different jelly bear formulations, which is important in assessing their acidity and potential impact on digestion.
Procedures for developing guava jelly bears for type 2 diabetes management. Formulation and preparation. Formulation A. Mix 49 grams guava juice, 49 grams water, and 2 grams of agar. Heat until agar dissolves, then solidify in molds. Formulation B. Combine 45 grams guava juice, 50 grams water, and 2 grams of agar. Heat until agar dissolves, then cool in molds. Formulation C. Mix 50 grams guava juice, 46 grams of water, and 4 grams of agar. Heat until the agar dissolves, then cool in molds. In vitro digestion procedures, oral phase, crushed jelly bears mix with alpha amylase and stir at 37 degrees Celsius for 2 minutes. Gastric phase, add pepsin and HCl to maintain pH around 3, stir for 2 hours. Intestinal, mix with bile salt and pancreatinine, incubate at pH 7 for 2 hours. Glucose estimation, use the Godpad method to assess glucose release.
This quantitative study on developing diabetic-friendly jelly bears from guava fruit followed strict ethical guidelines. Since no surveys, human participants, or testing on living subjects were involved, the research was conducted entirely through laboratory-based experiments. Researchers prioritized responsible ingredient and chemical handling, ensuring proper processing and disposal to reduce waste and environmental impact. Throughout the study, thorough documentation and transparency were upheld.
Our goal was to understand how these formulations break down in the body and how much glucose they release during the process. What we found is quite interesting. Formulation C consistently released the highest levels of glucose both during the oral and gastric phases of digestion. Formulation A followed, with formulation B releasing the least amount of glucose. This suggests that formulation C might be the most efficient in terms of glucose release, which could have significant implications for its potential use in diabetic-friendly snacks. This research is important because it provides valuable insights into developing healthier snacks for diabetics. By better understanding how different formulations behave in the body, we can aim to create snacks that release glucose in a way that is more controlled and beneficial for people with diabetes.
Our research analyzed glucose release during the gastric phase for 3 jelly bear formulations A, B, and C, each tested in 3 trials. Formulation C had the highest glucose release from ranging 381 to 388 mg per deciliter, suggesting higher sugar content or faster glucose release, while formulation B had the lowest, ranging from 300 to 315 mg per deciliter, indicating lower sugar content, while formulation A showed moderate release ranging from 325 to 338 mg per deciliter. From the study of Muller et al. 2018, glucose release during the gastric phase varies, with lower values indicating slower digestion and absorption. And according to Rajiv Hedda et al. 2023, as pepsin primarily breaks down proteins rather than carbohydrates, glucose release in gastric phase is lower than in the oral phase.
Continuing, let's take a look at the table 4. Formula C had the highest glucose release, from 352 to 402 milligrams per deciliter, while Formula B had the lowest, which is 304 to 315 milligrams per deciliter, suggesting slower sugar breakdown. Furthermore, Formula A showed moderate glucose levels from 311 to 365 milligrams per deciliter, indicating a steady breakdown pattern.
On the first table shows the average glucose concentration of the three formulations without enzymes, revealing slight variations. Formulation C had the highest initial concentration with 390 mg per deciliter, followed by Formulation A with 346 mg per deciliter, and Formulation B with 312 mg per deciliter. The total computed average glucose values showed lower concentration of the total glucose in Formulation A and B. The pH value is crucial for gelation and stabilization in confectionery production. The pH range of the guava jelly bear is 6.12 to 6.27, influenced by the ingredient percentages. The pH affects the gel strength, with lower pH weakening it and the higher pH increasing it.
In conclusion, the results showed that Formulation C had the highest glucose release across all digestion phases, Formulation A being the moderate, while Formulation B had the lowest. This variation suggests differences in sugar content and glucose breakdown efficiency among the formulations. The pH levels also varied due to different guava concentrations. This study supports guava jelly bears as a potential diabetic-friendly alternative to traditional candies due to their fiber and antioxidant content. However, limitations include reliance on in vitro methods, a small sample size, and financial constraints that may affect research accuracy. To improve the product, future studies should refine texture by adjusting the gelling agent, ensure controlled glucose release, and conduct human trials to validate findings. Expanding research funding would enhance accuracy and support further development of these jelly bears as a healthier snack option.
And that concludes our poster presentation. Thank you for listening.